All right, everybody, welcome to part 2 of our video. So we have at the bottom of our slide that there's 2 different types of capsids on this slide, but there's actually a third one. So we have the complex capsid, which is the last one. So let's take a look at these capsids. The first one is helical type capsid. So if you look at the lower left-hand side, you're going to see a naked helical capsid. So those nucleic acids are inside of the protein capsomeres. So those are on the outside. So it looks like this just little kind of rod or sandwich type structure. Again, they are teeny, 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 tiny. This is magnified 260,000 times so that you can see it, and it is that tobacco mosaic virus. On the right-hand side, then, we see this enveloped capsid. And if we go kind of like we did before with the bacterial cell inside out, you see that we have the nucleocapsid, or this nucleic acid on the inside wound up. Then we have essentially what we're going to call the capsomere, or the capsid. Capsomere, the repeating plates. And then we have the spikes poking through. And of course, this structure here, kind of a yellowish structure, is called the envelope. So again, the envelope is going to help with the docking and the uptake of the virus by the host cell. And so this is a different version of the virus. So it's a different perspective of that tobacco mosaic virus, because it then has that envelope. Okay. Icosahedral essentially is a three-dimensional shape, and it has these repeating plates. So there's 20 plates, so it gives us this 20-sided figure with 12 evenly spaced corners. So essentially, I mean, it looks like something out of science fiction, doesn't it? But essentially, we can see again that those spikes are sticking out, and very prominent, much more noticeable than an enveloped capsid. So, an enveloped icosahedral. So there's some examples there as well. So the complex capsid, literally to me, looks kind of like a cross between a mini robot and an insect. But essentially, this complex capsid is the type of structure for a type of virus called a bacteriophage. It is actually a virus that invades bacteria. And then they have essentially, again, that ability to convert that cell into something more dangerous, that bacterium into something more dangerous. They are never enveloped. You see that they do have the capsid head with the nucleic acid inside, and then they have this collar, a sheath, tail pins, I mean, kind of scary, and then these tail fibers. And essentially, those tail fibers are going to allow it to anchor, and then what it does is it injects that nucleic acid into its host. Okay, so you've already learned something about viral envelopes, essentially that it helps with the uptake of the virus into the host cell. There's other things to know, like where does that virus get its envelope, and it actually gets it from the host cell membrane. So when that piece of nucleic acid is ready to, or that pre-virus cell is ready to be expelled or released from a host cell, then it actually gets some of the cell membrane from the host cell in that process. Essentially, then the viral envelope is also going to allow for the packing of more viral proteins, and it hides the capsid from the host immune system. So it's kind of like being a little bit more discreet, that it just doesn't have those spikes hanging out, and it's easily identifiable by the immune system of the host. Yep, okay, so let's go on. Okay, we mentioned earlier that all viruses are going to have nucleic acid, either DNA or RNA or both. They have a few genes. You can see with the hepatitis B virus that they have 4 genes. Herpes virus has hundreds of genes, but still, this is far less than you would expect in a bacterial cell or in a eukaryotic cell. So it gives them the basic rudimentary information so that they can invade a host cell, redirect its activity, and then make more of itself. Of course, do the metabolism it needs to do or whatever else it needs to do. All right, so how viruses multiply. Essentially, what we see is a diagram showing us when this nucleocapsid is ready to be released from the cell. It's essentially moving up towards the inside of the cell membrane, and then it pushes out on that cell membrane. So part of the cell membrane is going to be wrapped around this nucleocapsid. And when it does that, then it's essentially released, and now it's on its own. Now you see this word virion. A virion is a mature virus that's ready to invade. Okay, so essentially, this cell cycle going on with the virus is going to continue to make more and more and more and more of itself. And yeah, so this is a different figure. This figure's from your book. And you can see that you have that nucleocapsid. This one has RNA in it. And then essentially it's moving up towards the membrane, and this nucleocapsid is going to get its viral protein matrix for the outside from the inside of the host cell. And so when it is being released through what we call budding, it's being wrapped around by that membrane with these proteins that are now functioning as the spike. The viral matrix protein functions as the spike. Okay, so we call this budding, and again, this is our mature virus or a virion. As mentioned before, this viral attachment to a host cell is very specific to receptors on the host cell. That may be beyond what you've heard in your courses before, but essentially all of our cells have receptors, and those receptors are going to dictate what can come into the cell. Very protective in nature. So viruses have the ability then, or they have evolved the types of envelope spikes that are specific to the host cell, so it responds to the host cell receptor. Okay. All right, I don't think you're going to forget this, right? Penetration by animal viruses. So essentially then looking at the figure from another figure from your book, it's showing us essentially that external, for this one, it's an enveloped virus, and that envelope and spikes then are essentially attaching to the receptors, right? And then it's being engulfed. So the cell membrane now is surrounding itself and bringing that virus directly into the cytoplasm of the host cell. And when inside the host cell, then that the envelope and then this membrane-bound structure or bubble we call a vesicle opens up and releases that nucleic acid directly into the cell. This one's showing us a free piece of DNA. Okay, so we know that viruses affect human health. We've all dealt with colds, maybe flu, chickenpox, one way or another. These are acute infections. So once you get exposed, then you have the infection pretty quickly after the initial exposure. Herpes and warts. So certainly there's a wide variety of viruses that we fight off with common everyday warts, as well as, genital warts. Prominent viral infections worldwide, there's a list there. A big one, yellow fever, which if you're going to travel to some countries, like we went to Tanzania, Uganda, and Rwanda last summer, we had to get a yellow fever vaccine. So you have a yellow card, which is a vaccine card. We know that there's a number of seriously high mortality rates associated with viruses. We all remember COVID-19. We've heard about AIDS or HIV, Ebola, and rabies. Infections that cause long-term disability. So we've heard that with COVID-19, with the long COVID, of course, polio, and I mentioned neonatal rubella. Another thing that we know about viruses is that viruses are connected to some chronic diseases, including a variety of cancers, potentially Alzheimer's, obesity, and type 1 diabetes. For our infections, so remember how it said acute infection. Well, we have some infections that are persistent infections. So essentially they can last, from weeks to the whole lifetime of the host. And they just kind of hang around